we had finished an introduction to bacteriovirus, bacteriophage replication. And we said that it really serves as a model for us to understand the replication of viruses that infect animals and infect humans. And furthermore, you guys, again, just such exciting news that we're now renewing interest that originally developed in Russia before antibiotics were discovered. So it was in Russia, in the former Soviet Union, that scientists explored the use of bacteriophage to kill bacterial pathogens causing infections. And then with the discovery of penicillin and all our other antibiotics, people lost interest in it. But now we have these multi-drug resistant, multi-antibiotic resistant bacterial pathogens. They're actually having some really good success treating those multi-drug resistant bacteria with bacteriophage. So again, folks, I hope you have a chance to at least skim this article, because I think this is going to be a powerful tool in our arsenal, right, to help combat some of those multi-antibiotic resistant bacterial pathogens. Okay, so folks, where we're going to go then in our PowerPoints, we're on the virus intro, and we're going to start on slide 40, and this is our introduction to animal or human viruses. So we might wonder, why did our understanding of virus replication in animals or humans, why did it lag behind our understanding of bacteria? And earlier, the only way that people could study the replication of animal-human viruses was to actually inoculate humans or animals. And even though there's been some absolutely horrible experiments you guys done using people as guinea pigs, slowly, hopefully, we're getting better. So that there were moral and ethical issues about purposely inoculating animals to see these viruses replicate. So one of the advances was it was discovered that some viruses, some animal and human viruses, can replicate in embryonated eggs, fertile eggs. But it's a bit challenging, folks, because not all viruses will replicate in the eggs. It takes some expertise because you actually have to inoculate different viruses into different parts of the embryonated egg. There's always the challenge that the eggs could get contaminated, right? But it was better than inoculating humans or animals. And then, folks, there was this huge breakthrough in the 1950s when a new technique was developed called tissue or cell culture. And I'll just describe it, folks, really crudely. And I'll describe it how it was done in the old days, like when I was going to school. So in tissue or cell culture, you'd have a sterile tissue or culture dish. And what you would do is you'd take cells, like maybe you could take, say, a piece of liver or a piece of kidney, and you would grind up the solid piece of tissue in a grinder to separate the cells, and then you put it into your tissue culture dish and add an appropriate medium, food for your cells. And if you were lucky, the cells, this would be the medium here, if you were lucky, if your cells could survive and use the medium for food, they'd start dividing. And what you would get would be a monolayer of cells. So this is a monolayer of cells. And this was so awesome because now viruses could be introduced, and you would hope, keep your fingers crossed, that when you introduced a virus, it would be able to bind, so this will be our little virus, it would be able to bind to the cells if they had the appropriate host cell surface receptors and infect the cells and then replicate in the tissue culture cells. So you weren't inoculating a human or an animal. And the idea was that they would replicate and infect other cells, right? And eventually you'd get lots and lots of virus in this medium. And then what was really cool is then you could maybe have like 10 tissue culture cells and infect them with viruses, and then add different drugs, different chemicals, to see if, could any of the drugs stop the replication. So this would be a cool way to screen drugs for antiviral activity. Another purpose was you could harvest the viruses and use them to make vaccines. And you guys, it's amazing. We still, well, no, okay, I will. Okay. But one of the problems is, folks, is with normal cells will only divide a certain number of times. And from A and P, you probably remember that in, for example, human cells, we have these linear chromosomes. And so every time our chromosomes are replicated, we lose little bits at the tips. And so we have these special DNA sequences called telomeres at the end of our chromosomes. And every time our chromosomes replicate, the telomere gets shorter and shorter and shorter until eventually, right, the telomere is all used up. So if the chromosome is replicated, now we're starting to lose coding DNA sections, right? And that cell can't divide anymore. So that meant that these tissue cell lines, you could only pass them maybe 30 or 40 times. And in science, when you're trying to do repeat experiments, you want everything to be the same, right? So this is a kind of a disadvantage. So what folks really wanted, what was desired was what are called immortalized cell lines. Immortalized cell lines. What do you think an immortalized cell line would be, you guys? How would they differ from normal cells? They just keep dividing and dividing and dividing and dividing. And what does that sound like? Cancer cells? Yeah, right? So cancer cells. And so, folks, this is really, really a heartbreaking story. And, okay, before I get to the heartbreaking story, this is just a diagram, folks, of our embryonated, our fertilized eggs, and then showing all the different places that different viruses would have to be inoculated to get them to grow. And again, folks, it takes expertise. There's always the danger that eggs are going to get contaminated, and not all viruses will grow in embryonated eggs. One thing I wanted to share with you is that the viruses used in the inactivated influenza vaccines, they are grown in embryonated duck eggs. So when you go to get an influenza vaccine, do they ask you if you're allergic to eggs? Yeah, they do. And why is that? Because the vaccine could contain some of those antigens or allergens from the eggs, right? And so it could trigger an allergic response in you. So that's why they ask, are you allergic to eggs? Right, okay. And then, folks, like the little cartoon I showed you there, that was back in the dinosaur days. Now they have these tissue culture containers that are of soft plastic. And again, the cells are going to be grown inside. This is the medium. It usually has a pH indicator in it to indicate if the pH is getting off. And then you can see these ports where you can inoculate your viruses. You can inoculate different drugs, for example, to see if they could act as antiviral drugs. You could harvest viruses, maybe to do vaccine studies. But again, one of the problems was that these cell lines, they kept dying out, after 20 or 30 passes. So, folks, and you probably have heard the story, The Immortal Life of Henrietta Lacks. Okay, now, back in my day, we were told, we have discovered this immortalized cell line, and they're called HeLa cells. And back in the day, we said it stood for Helen Lane. And you guys, that was totally wrong. It's not Helen Lane. And I apologize, you guys, in the supplemental notes, I did the name wrong, and I so apologize because I so respect the person. HeLa cells came from Henrietta Lacks. And this is really kind of the downside of people being morally and ethically not good. So what happens was Henrietta Lacks was a young mom. I think she had 4 little children, and she developed cervical cancer. And what happened was she died. It eventually killed her. But what the doctors, scientists did was they harvested her cervical cancer cells and discovered that they were what? What were they after? An immortalized cell line, right? They kept dividing and dividing and dividing and dividing and dividing and dividing. But what was unethical about it, folks, they didn't ask her permission, nor did they ask permission of her family. And these cells were used, they've been used in labs all around the world. We'll see you guys, a scientist won the Nobel Prize for he discovered that within the cervical cancer cells, there was a human papillomavirus DNA had inserted. So he got a Nobel Prize for that because he was showing the connection between some viral infections and cancer. He got the Nobel Prize for that. And meanwhile, the family had no idea, right, that these cells were being used. So people got awards, they got money, companies use them for vaccine development. And my understanding is, folks, I haven't read the book, I'm embarrassed to say, but my understanding was it wasn't until this journalist, Rebecca Skloot, decided to pursue the story of, where did these cells come from? What was the story? That the family, it was only when the journalist was doing the investigation and finally tracked it back through the medical records that the family was made aware that their mother's, grandmother's cells were being used in all these labs. So anyway, not a good chapter in science history. But these HeLa cells, again, folks, they've been really powerful in poliovirus studies and doing HIV studies. They're still being used in labs around the world, and I don't really know if the family now is being able to share in the profits that have been made from using these cells, right? But it's just, like I said, kind of a bad part of our history. Okay, so, and we might ask you guys, in these cancer cells, why is it that they keep dividing? Well, they have telomerases, enzymes that keep lengthening the telomeres, and that's what permits them to keep dividing over and over and over again, yeah. And again, folks, you might say, well, what does this have to do with microbiology? Well, again, we're going to discover when we talk about human papillomavirus, some of them can cause our cells to become cancerous, and that's exactly what happened with Henrietta Lacks. She had a HPV strain 18 infection, and the HPV virus basically, redirected her cervical cells just to divide as cancer cells. And indeed, you guys, when they do analysis on cervical cancer cells from women, I think at least 80% of them show infection with specific strains of HPV. So we'll talk about that, right? We'll talk about that. We'll come back to it. Okay. There was a movie done, folks, on this, and I think Oprah Winfrey, played the part of one of the grown children of Henrietta Lacks. Okay, yeah. All right, folks, so with kind of that introduction then, what we want to do is very superficially look at animal-human virus replication, and we're going to see how many of the stages are really similar to the bacteriophage, all right? So, folks, with a virus infection, so human-animal virus infection. What's always the first step, folks? If we're at the cellular level, what's the first step? What does a virus have to do to invade a cell? Attachment, right? There you go. What's a fancy term? Adsorption, yeah. And we'll see you guys, if we can block attachment, if we can block absorption, you're not going to get sick, you're not going to get infected. We'll come back to that. Okay, so folks, amongst the human/animal viruses, there's 3 strategies for entry. And entry, you guys, is the second step, right? So what do we mean by entry or /penetration? What's the goal here? Yeah, how does the viral genetic information get into the cell, right? And there's 3 ways that can happen. How does viral genetic information get into the cell? And remember, folks, in animal cells, there's no cell wall, right? There's no cell wall, not like a bacterium. So, folks, and you do not have to remember which viruses use which entries, just to give you an overview of the strategies. So, folks, with this is called direct penetration. This seems really similar to bacteriophage entry, because in this case, and one example is poliovirus, the poliovirus attaches to those specific host cell surface receptors here indicated by these little pink discs. This is the surface of the human or animal cell. The virus attaches by its adhesins to the host cell surface receptors, and then using proteins forms a pore in the cell membrane. And it's through that pore then that the virus introduces its genetic information into the host cell. And again, you guys, this is a little bit like bacteriophage, right? Because remember, the capsid remains outside, and the bacteriophage just injects the bacterium with its nucleic acid. So direct penetration looks a lot like bacteriophage entry or penetration. This example, folks, this is a really interesting example, and this is called membrane fusion. And this is the approach that HIV uses. Okay, so with membrane fusion, you guys, we're always starting with an envelope virus. And folks, remember, where did the envelope come from? It's stolen host cell membrane, right? And remember, folks, the cell membrane, the fluid mosaic model, phospholipid bilayer, the consistency of olive oil, right? Okay. So with HIV will have adhesins in its envelopes that bind to specific surface receptors on our cells. And then, folks, HIV has an envelope protein that triggers fusion of the HIV envelope with our cell membrane. It's like the HIV envelope just melts into our cell membrane, right? Because remember, the envelope is just cell membrane itself. Yeah, just think of like olive oil, melting into another droplet of olive oil. And when the fusion is completed, that permits the release of the capsid carrying the genetic information into the cytoplasm. And in the cytoplasm, then, those little protein subunits, the capsomeres, they're disassembled, right? That's called encoding. And then the viral genetic information is released, right? So again, I'm hoping folks will have a chance to come back and walk through all the steps of HIV replication. But again, membrane fusion. There is an anti-HIV drug that helps interfere with the protein that's involved in membrane fusion, right? So that's hopefully a step where we can block the ability of HIV to get its genetic information into our cells. And then, folks, this last one, endocytosis, this is the process used by influenza virus, which I also will have a chance to talk about. And to me, it's almost like the Trojan horse approach. So remember when the Greeks were attacking Troy and they couldn't beat them, so they said, okay, let's pretend we're leaving and we're going to build this big wooden Trojan horse and leave it outside their gate as a farewell present, right? And so the Greeks left the Trojan horse there and they pretended to sail away. And then the people of the walled city opened up the gates and said, oh, they left us a present, and pulled the Trojan horse into the city. Well, what happened, you guys? What was inside the Trojan horse? The Greek soldiers, right? So they were able then to defeat Troy. Yeah. Okay, so influenza must have read that story. What was that story, you guys? Was that the Iliad? Okay, viruses don't read. So here's your cell, and let's pretend this is influenza virus, right? Okay, so influenza virus is an envelope virus. The envelope adhesins bind to our host cell surface receptors, and it triggers our cells to say, oh, a present. And so what's happening right here, you guys? Endocytosis, right? Our cell actively participates to bring the influenza virus into the cytoplasm in a membrane-bound vesicle. And it's once it's inside this vesicle then that the influenza proteins trigger fusion of the envelope with this endosome membrane, releasing the capsid, they're encoded, and then the viral genetic information is released. So again, folks, like 3 different strategies that viruses can use to get into our cells. Yeah? And you don't have to remember who does what unless we come back and talk about it in greater detail. Okay? All right. All right. And then, folks, so we have entry penetration, and then we would have biosynthesis. Right? So this is when the viral nucleic acid is replicated. And viral proteins are made. And who's going to do all that work? Yeah, the host cell, right? So this is going to be done basically using the host cell biosynthetic machinery. Sometimes using the host cell, maybe DNA polymerase is certainly using the ribosomes of the host, right? And this, folks, is where it gets a little bit messy. Okay. So first, just a general overview. So in general, folks, most RNA viruses are going to be replicated in the cytoplasm, right? And in general, most DNA viruses get replicated in the nucleus. And to me, this makes sense, you guys, because let's say it's a DNA virus. Which enzyme does it need to copy its DNA? DNA polymerase, and where would that be in our cells? In the nucleus, right? Yeah. We're going to see, you guys, with RNA viruses, they have to bring, they either have to encode the information for their own RNA polymerase, it's going to be a special RNA polymerase, or they bring pre-made RNA polymerase with them, and we'll explain why they have to do this, okay? There's always exceptions. Don't worry about the exceptions, you guys. So after synthesis, just like in bacteriophage, there's assembly, and then there's going to be release. And again, folks, the viruses have different strategies for being released. So the easiest way is if the viruses cause the poor little host cell to just lyse, just break open, right? So just like in the bacteriophage lytic cycle, right? And again, you guys, this is going to contribute to a lot of damage to our tissues, right, our cells. But another way that some viruses can be released is the opposite of endocytosis. They'll be released in the process called exocytosis, right? And then to me, folks, this is such a cool way. Many of the envelope viruses will be released through a process called budding. And this is where we can see how they're going to steal our host cell membrane. And folks, one reason I like budding, well, it's how HIV escapes from our cells, and it's also how influenza viruses escape from our cells. And it helps us understand how they acquire their envelopes. They're going to acquire the envelope by stealing our cell membrane. And indeed, you guys, here's a little cartoon of a envelope virus that's ready to be released through the process called budding. So folks, here's the capsid, right? So the capsid is assembled, the viral genetic information has already been packaged. Now, the virus is going to steal this part of the cell membrane. What must it insert into the cell membrane before it steals it? Yeah, viral proteins that will act as adhesins, right? At the very least, the virus has to get its viral proteins into the membrane. At the very least, get the viral adhesins in there. And folks, we'll come back to this when we do immunology. Our immune system can detect those viral proteins in our cell membranes, and that will target our cells for what? Those of you that have had AMP. For destruction, right? Those viral proteins in our cells tell the immune system this cell has to be destroyed. So part of the damage we see in viral infections is a result of what? Our immune response. And you guys, this is really common with viral infections. A lot of the damage is done by our immune response, right? We talk about a double-edged sword. Our immune system is a double-edged sword. Like we're using it to fight the invading, pathogen, right? So here's my double-edged sword. And one side is sharp, right? Because I want to kill the invading pathogen. But the problem is how I imagine you guys, the other side is sharp. And when I whack at the pathogen, I might get this rebound and hurt myself. And indeed, you guys, our immune system, we can't live without it, right? But it also contributes to a lot of damage in many infectious diseases, especially viral diseases. Okay, so folks, here again we have the virus that's is getting ready to bud, inserts its proteins in our cell membrane, and it's so wild, you guys. If we could watch it with an electron microscope, it looks like the capsid goes up and starts pushing against the cell membrane, right? And eventually we'll have this evagination. And eventually the virus will escape, stealing a layer of cell membrane that will become the envelope, right? And then it can go and just travel to the neighboring cell and bind to it and start the cycle all over again. Pretty amazing, pretty amazing. Now folks, this is where it gets kind of messy and I apologize. But what we want to do, folks, is back up here to biosynthesis and talk about what kind of enzymes will different viruses require to copy their viral genetic information, okay? And let me just go fast forward here really quick, you guys, to see. It might be the next slide might act better. Okay, so you guys, I'm going to use this as my kind of my guide here on the board work, because I think this really is kind of to the point. This is what I want you to know for the exam. So I'm going to ask you a bunch of questions. And of course, these would be delicious exam questions. Okay. So let me make a table, guys, and I'll ask the questions and we'll fill in the table. Okay, you guys, so the first question I want to ask you is let's talk about cells. Okay, let's talk about cells, right? And if, so cells, your cells are bacteria cells. So when cells need to copy their chromosomal DNA, which enzyme do they use? Good job, you guys. DNA polymerase, right? All right. So with cellular DNA polymerase, folks, what's the template? What's the guide? And so you can look at the table here. It's totally cool. Single-strand DNA, awesome, you guys. And what's the product? Single-strand DNA, right? Complementary DNA. Does that make sense? Okay. Now, folks, because things are gonna get messy with our virus enzymes, we need to have a fancier way of naming these enzymes. Instead of saying just cellular DNA polymerase, I'm going to use a naming convention where I express the template and the product. Okay? So the fancy name, you guys, for cellular DNA polymerase is DNA dependent. This is telling me what the template is. And then the second part of the name is what's being made. So you guys, what am I making with cellular DNA polymerase? DNA, right? So it's DNA-dependent DNA polymerase. Okay, so far you're like, well, we don't quite understand why you have to do this, but we'll get there. Okay, so now let's look at cellular RNA polymerase. So folks, what is the template for cellular RNA polymerase? And again, you can look at the table. It's totally cool. Single strand DNA. Good. And what is the product? What does RNA polymerase make? Good. Single strand RNA, complementary RNA. So you guys, using my naming technique, how would you name cellular RNA polymerase? DNA dependent. That's the template, right? Good. DNA dependent. And the second part of the name? RNA polymerase. Awesome, you guys. Excellent. Okay, now here is where things are going to get messy. Okay, so you guys, so let's think about this. Let's pretend we're an RNA virus. We are an RNA virus. What is your genetic information? RNA, right? So, if you need to make copies of your RNA, what is the template? If you're an RNA virus and you need to make copies of your RNA, what's the template? What's the guide going to be? RNA, right? So if you get into, say, a human cell, is there any enzyme in that human cell that can use RNA as a template to make RNA? Nope, right? So that means RNA viruses have to encode information for a special viral RNA polymerase. So you guys, let's just call it, we'll call it a, I'm going to put RNA virus, RNA polymerase, right? Because I want to emphasize these are RNA viruses. Okay, so you guys, if you're an RNA virus, what's going to be the template for making copies of RNA? RNA, right? Single-strand RNA. And what's going to be the product? Complementary single-strand RNA. So you guys, how will you name the RNA virus RNA polymerase? There, you got it, you guys. You nailed it. And again, folks, host cells don't make an RNA-dependent RNA polymerase. So the virus has to carry the genes for it, right? Get the host cell to make it, right? Because the host cell doesn't make it. Does this make sense, you guys, so far? Okay. And then, you guys, we're going to blow you out of the water. Crazy, crazy viruses. So there's a special group of viruses. You guys know these already. They're called the retroviruses. Let me put it over here, you guys. So retroviruses, they're a special group of RNA viruses. And you guys, why are they called retro? What does retro suggest? Backwards, right? So remember, you guys, what the RNA viruses do, they drive information flow backwards. So we're going to have HIV RNA, right? And the HIV RNA is going to act as a template to make what? DNA. Isn't that just bizarre, folks, right? So this is the template, and we're going to make what? Make DNA, right? So you guys, so let's do that here. What's the, oh, sorry, you guys, what's the name of the enzyme that does this? So bizarre. Yeah, the enzyme is called, let me put it in red. It's called the HIV, do you remember the name, you guys? Reverse transcriptase, right? We're driving transcription backwards. And indeed, you guys, this was the first target for the first anti-HIV drug. AZT targets the HIV reverse transcriptase. And folks, it actually turns out it's a two-step process, and I still cannot believe this. So if you guys say, no, it's not possible, I agree. So you guys, in step one, okay, we're gonna do our HIV reverse transcriptase. And on the exam, you guys, it would be the first step that I ask you about, step one, because this is the bizarre one. In step one, what is the template? RNA, good. And in step one, you guys, what's the product? What are we making? DNA, good. And again, this is step one. So you guys, in step one, how would you describe the HIV reverse transcriptase? RNA dependent. And what's it making? DNA, yeah? Crazy, crazy. Yeah. And folks, I will explain step two, but I'm not going to put it up here because this is a step I want you to remember, this bizarre step where RNA is a template to make DNA. The second step, you guys, again, unbelievable. Reverse transcriptase can okay, so you guys, so we start out with HIV RNA, right? So there's our HIV RNA, and what's reverse transcriptase going to make? DNA, right? And this is the bizarre step, you guys. Then the reverse transcriptase, it comes in and it removes the RNA strand, it hydrolyzes it, and then uses the DNA as a template to make what? DNA. And you guys, I will not ask you about that on the exam. I'm like, you've got to be kidding. That's the most ridiculous thing I've ever seen, right? But you guys, just as a little appetizer for discussion of HIV replication, this double-stranded DNA, we're going to call the HIV provirus. What does that sound like, sort of like? Does it sound a little bit like a prophage? And where do we find prophages? In chromosomes, right? Where do you think the HIV provirus is going to end up in us? In our chromosome. And once it gets in there, we can never get it out, right? So this HIV provirus then will be inserted, it inserts into the human chromosome. And we can't get it out, ever. Never, ever, ever. Okay. So again, you guys, just a little appetizer. I hope we'll have enough time to walk through all the steps of HIV, but it's just so like, certainly, I mean, it seems like it comes from another universe somewhere, right? Just violating all the rules, all our rules. Okay, all right, you guys, so you guys, this is delicious, delicious exam type question, okay? Let me just quiz you really fast, you guys. Cellular RNA polymerase, what's the fancy name for cellular RNA polymerase? DNA-dependent RNA polymerase. Good job, you guys. What's the fancy name for cellular DNA polymerase? DNA-dependent DNA polymerase. What's the fancy name for an RNA virus RNA polymerase? RNA-dependent RNA polymerase. Good job, you guys. And in step one, what's the fancy term for HIV reverse transcriptase? RNA-dependent DNA polymerase. You nailed it, you guys. You nailed it. Good job. Okay, that was awesome. You guys got that. Okay, just one last little tidbit, folks. So, so it turns out when we're talking about RNA viruses, would you guys be okay if I erase this? Is that okay if I erase this? And we've got it on the PowerPoint slide there. Okay, just one last little tidbit, and I'll try to make it really exact, hopefully. Okay, so with RNA viruses, folks, you can have single-strand RNA virus. So they only have one, one strand of RNA. And then you can have double-stranded RNA viruses. Okay. And it's like, well, what's the point? Well, you guys, let's take a look at a double-strand RNA virus. Because it turns out, okay, so this is an RNA strand, and this is an RNA strand. It turns out there's two different types of single-strand RNA, and what I want you to remember, you guys, is what's called positive sense single-strand RNA. This acts as mRNA. What does that mean, you guys, that it acts as mRNA? When that positive sense RNA gets into the host cell, what can bind to it? What structure in our cells can bind to positive sense RNA? Ribosomes, you got it, right? So positive sense RNA acts as mRNA, right? Host ribosomes bind and translate. Translate, right? Going to make the viral proteins. And you guys, I know you're not going to be happy with me. But if you can just remember that for the exam, that's all you're going to need to know about positive sense RNA. So this, we'll use a plus sign, and then the complement is negative sense. And you guys, it's like on the exam, what am I going to ask you? This positive sense single-strand RNA acts as mRNA. That is almost exactly the question I'm going to ask you. And the reason, folks, I don't want to go into the negative sense because that's a whole lecture in itself. Okay, so if you can remember that positive sense RNA acts as fill in the blank, mRNA, you nailed it. Okay, if we had many lectures, you guys, on viruses, we'd go into the details of viral RNA replication. It gets really, it can get really complicated, and for us, it's not going to serve any purpose for our class. Okay, so we're just going to leave it. And I think you guys, let me back up here. Yeah. So you guys, the table that I skipped, I put a star there because that's what I'm going to ask you about positive sense RNA, that it acts as messenger RNA. Okay? Great. Okay, guys, so we got through, that was really the hardest part we just did. Okay, so hopefully the rest of it, it'll be a little bit easier for us to digest. So folks, just like, before we said when you're talking about bacteriophage, we said they're lytic phage, right? And then there's temperate phage. So we can classify the viruses in somewhat similar terms. So there's some viruses, you guys, that are always going to lyse the host cell, right? So what would you call them? Lytic viruses, right? And another term that some folks use is cytocidal. What's the suffix cyto mean? To kill, right? And what's cyto mean? Cell, right? So cytocidal viruses, right? They're always going to cause lysis of their host cell. They're always going to kill the host cell. And of course, I tried to find the most dramatic pictures possible, right? So this is an adenovirus, a naked virus. It's probably one of the causes, you guys, that are causing a lot of our upper respiratory tract infections. It's naked like a cold virus, so it remains infectious for long periods of time. And this is really dramatic, but it can cause lysis of cells, leading to this blood red. The white part of the eye is a sclera, right? AFP folks, yeah, right, almost like a vampire or something, right? Okay. And folks, don't worry about the graphs. And indeed, this might be like a little nerdy bit of virology, but if we were watching like a a lytic or cytocidal virus, maybe like in our tissue cell culture, and we were taking samples of the medium and trying to count the number of viruses present. Like when we initially infect the cell culture line, we're taking samples of the medium, it's like there's no viruses present. Where did they go? Where are they? They're in the cells, replicating, right? And then all at once, the infected cells break open and release massive numbers of viruses, right? So you'd see a big spike in the number of viruses released. So that's classic for a lytic virus, right? But folks, there are other viruses like HIV that won't kill the host cell right off the bat. What they'll do is they'll just bud and bud and bud and bud. Or another great example, you guys, is hepatitis B virus, right? When our liver cells get infected, what do? They bud, and they bud, and they bud. And eventually the host cell will die, right? The host cell is exhausted, it can't take care of itself. All it's doing is making more virus. Eventually the host cell dies. But what this means is that instead of, like if we were doing a tissue culture study, instead of seeing a big spike in the number of viruses, right, when all the host cells are lysed and die, we see this slow, gradual increase in the viruses, right, as they are slowly released from the host cells. The reason this is important, you guys, in these persistent infections and with HIV and hepatitis B, it means that you can end up with really high loads of virus in your body because they're constantly being released from your infected cells, right? So with hepatitis B virus, you can have incredibly high levels of virus, like 10 to the 8 virus per milliliter of blood. You guys, that is crazy. Really, really high levels of that. So, again, you guys, kind of nerdy. I probably wouldn't ask you, what's the difference between the lytic release and the persistent budding, but just something for us to keep in the back of our mind when we're talking about the different viral pathogens. Now, you guys, this is a great topic I would like to ask you, okay, and this is going to kind of be an appetizer for immunology. And so, you guys, I'm going to use this cool human-animal virus replication poster. Did we talk about this already, you guys? What did they use for the viral adhesins? Yeah, I just love stuff like this. I'm easily amused. Okay, so you guys, so this is the surface of the host cell. What are the little green balls? Those are the host cell surface receptors, right? So to infect, what does the virus have to do? It has to attach, right, to those host cell surface receptors. Step one, right? Well, you guys, if we can somehow physically block, right, the viral adhesins from binding to the host cell surface receptor, would that block infection? You better believe it, right? That's exactly what your immune system does, folks. What your immune system does is your immune system is going to make antibodies or immunoglobulins against those viral adhesins. And you guys, thank heavens, viral adhesins are made out of proteins. Proteins are the best antigens. They trigger the strongest immune response, okay? So in this particular example, folks, okay, so we'll put our little virus model up here, okay? So historically, you guys, what we use is our cartoon for antibodies or immunoglobulins. They're made out of proteins. We historically cartoon them as little Y-shaped molecules, right? And those of you in A&P, you probably know at the ends of these little arms, there's special, what they call antigen binding sites, right? And what are they going to bind to, you guys? In this case, right, if these are antibodies, bind, what do we want them to bind to, you guys? The viral adhesins, right? So, see, I need more toys. I need some antibodies here. But you guys pretend my hand is the antibody, right? And pretend I'm an antibody directed against the viral adhesins. So check this out. I'm going to bind to the adhesins, and now can the virus bind to my host cell surface receptors? Nope. And you guys, there's a fancy term. Why they make these fancy terms, I don't know. But there's a fancy term for antibodies which block attachment. Excuse me. And it could be attachment of a pathogen. It could be blocking attachment of a toxin. Antibodies which block attachment are called neutralizing antibodies. And again, folks, the reason the neutralizing antibodies are so powerful, if you block attachment, you block colonization, you block infection, right? You block the toxin getting into your cell. And indeed, you guys, this is an important arm of our immune system, and it's also one of the goals of which process that humans have developed? Vaccination, yeah? So you guys, this is why vaccines are so powerful, is they trigger production, right? The vaccines trigger production of what kind of antibodies? Exactly, neutralizing antibodies, you guys. You block attachment, you block disease. Isn't that amazing? So that's a big goal, a big accomplishment of many of our vaccines, is they're going to trigger your immune system to make neutralizing antibodies against that particular pathogen, against that particular toxin. Isn't that awesome? And isn't it elegant, you guys? That's what blows me away with nature. It's so elegant. So in this cartoon, folks, this is a cartoon, and obviously I'm not going to make it in a cartoon studio. Here's my host cell, and what are these little cup-like structures? The host cell surface receptors, right? So you guys, let's first tackle this virus. What are these lollipops, you think? What function would they have? Those are the adhesins, right? So you guys, what's binding to the adhesins? Antibodies. What kind of antibodies? Neutralizing antibodies. Can this virus then attach to your cell? Nope, not going to attach. And folks, again, just to give you an idea, these neutralizing antibodies, they can protect us against toxins. We're going to see the protein toxins. They have to bind to special cell surface receptors. So if we can coat them in neutralizing antibodies, they can't bind to our cells, and they're not going to get into our cells, and therefore they're not going to cause us harm. This is what the tetanus vaccine does for us. It makes neutralizing antibodies against tetanus toxin. It's what the diphtheria vaccine does. Produces neutralizing antibodies against diphtheria toxin, and I'm forgetting of DT, diphtheria, tetanus, pertussis, right? Pertussis and whooping cough, the same thing. The vaccine is going to produce neutralizing antibodies against the pertussis toxin. Isn't that awesome? And indeed, you guys, with bacterial pathogens, many of them have to attach to our cells. Same thing. We make neutralizing antibodies against them. The antibodies bind the adhesins. The bacteria can't bind to our cells. They can't colonize. Yeah, isn't that gorgeous? Your immune system is amazing, amazing. Okay. And folks, this is one of the study guide questions, and this is like, why, it seems like we have all these antibiotics to treat bacteria, right? And it seems like, indeed, if the bacterium isn't resistant to a lot of antibiotics, usually we can find an antibiotic that will inhibit or kill the bacterium. But folks, why is it so much harder to treat viral infections? Okay, so we want to remember, you guys, who is replicating the viruses? We are, right? The viruses are using our biosynthetic machinery, using our metabolic machinery to make copies of itself. So you guys, if we stop viral replication, could we also harm normal function of our cells? Yeah, and that's why very often the antiviral drugs we take, they have some really not very pleasant side effects, right? Because they're actually interfering with normal function of our cells. One of my friends has had HIV for a long time. He's on a good mixture, what they call hard therapy, highly active antiretroviral therapy. It's what we used to call a cocktail, right, where you have multiple drugs. And so he was doing well, and then he had an HIV breakthrough, developed resistance, right? So they had to change his drugs, and he said it was horrible. He was having nightmares and hallucinations, right? So that's an extreme case, but we do want to remember, guys, often the antiviral drugs, they are interfering with normal cell function, and therefore a lot of times you have some really unpleasant side effects. Yeah, so it's just bad. And furthermore, you guys, where are the viruses replicating? Where are the viruses replicating? Inside your cells, right? And you guys remember, let's see here. Okay, so if this is the human cell, right, and you're taking an antiviral drug, how do you think the concentration outside your cell compares to the concentration inside your cell? Inside is probably going to be what? Lower, right? And why is that? Right? That phospholipid bilayer, right? Is that like a little diffusion barrier? Yeah? So sometimes it's hard to find drugs that you can reach therapeutic levels of your antiviral drugs inside the cell without causing harm to the cell, right? So lower antiviral drugs. This is another problem, you guys, is being able to concentrate the antiviral drug inside your cells at high enough concentration to stop replication of the viruses without doing what? Harming your cell, right? Yeah, what a pickle. What a pickle. Okay. And folks, again, I won't go into this much other than I think there's so if we back up, you guys remember how in bacteria we're trying to find unique bacterial targets for our drugs, right? So we're going to do the same thing with our viruses, folks. And in HIV, can you think of a unique target? What does HIV use that we don't use? Reverse transcriptase, right? So that's an awesome target. So the HIV reverse transcriptase, that is an awesome target. And indeed, you guys, this was the first target of the first anti-HIV drug. Do you guys remember what that was? AZT. Yeah, I remember it because I remember when this, when they first discovered it, and we were just, oh my God, we were so sad. So you guys, so AZT is what's called a nucleotide or nucleoside analog. It looks enough like the normal nucleotide that, for example, reverse transcriptase will incorporate it into the strand of DNA. But these nucleotide or nucleoside analogs, either they cause incredibly high mutations, right? They base pair incorrectly, they cause lots of mutations, or they cause what's called premature chain termination, right? It's like once they get incorporated, an incoming nucleotide can no longer be added. So either through really, really high mutation rates or premature chain termination, you stop the virus from replicating. And you guys, did AZT originally work really well? Oh man, it was awesome, right? It was awesome, you guys. But we're going to discover with HIV, let me pull this down, you guys. But the bad news, you guys, with HIV is its incredible genetic diversity. And there's 3 sources of it to help us understand why it mutates so rapidly, why it rapidly evolves drug resistance. So first of all, folks, the HIV reverse transcriptase doesn't proofread, right? So now we're going to start introducing lots of mistakes. And then, folks, remember how we said that once the HIV provirus is made, it gets stuck into our chromosome. Remember that, you guys? Okay, so when the cell is going to start becoming an HIV factory, it's the cellular RNA polymerase that transcribes the HIV provirus into HIV RNA. And do RNA polymerases proofread? Nope. So then the RNA polymerase, it's the same story. So you guys, so there's 2 steps in HIV replication where we're going to introduce lots and lots and lots of mutations. And you guys, even to make this worse, it turns out that in HIV, there's 2 strands of the single-strand RNA genome. And what this means is, and we're going to see this again in influenza virus, a single cell can get infected with 2 different strains of HIV, right? And so when we have assembly of the HIV, right? It can grab an RNA strand from this mutant strain and an RNA strand from that mutant strain and package them together. And that's almost like us, sexually reproducing organisms, right? We get genetic information from mom, genetic information from dad, right? In a weird sort of way, that's what HIV is doing, right? Okay, so the two strands may come from different HIV mutant strains. And for this reason, you guys, when at first we were so hopeful that AZT, I can do this. We were so hopeful that AZT, maybe, right, we'd be able to start saving lives. What happened? Yeah, quickly, HIV evolved what? What did HIV back in the old days, you guys? It quickly evolved resistant to what? To AZT, yeah. And nowadays, folks, we know you never treat a patient with HIV with a single anti-HIV drug. You always use combination therapy, right? Because if you're only using a single drug, what's going to happen within a few months? You'll have a mutant evolve that'll be resistant, yeah. So you always are doing combination therapy with it, right? And again, folks, I'm hoping we'll have enough time that we can walk through all the good news, you guys. Now they have drugs to block almost every step in HIV replication, right? And that's, oh my gosh, you guys, that is really good news. That's something for us to be hopeful about. But what do we really need? A vaccine, right? And this is the problem, you guys. All this genetic variation going on, we can't find a vaccine that will protect against all the strains. Yeah.
You wrote rapid resistance to any
AZT, sorry you guys, the AZT, this is our, and you wouldn't have to recognize the structure, you guys, but it's a base analog to thymidine, right? Thymidine, so when it gets incorporated by reverse transcriptase, I think it actually just stops the nucleic acid synthesis, so it's going to stop the HIV provirus from being made. Yeah, okay, does that help? My chicken scrawls here. Okay. Okay, now folks, now we're going to get into some talking about some of the different consequences of viral infection. And let me see, you guys, if I can find on, I'm so bad about, I give you these handouts and then I don't refer to them. So I did tell you guys there was actually a table there that had all the, anyway, okay. You got a table there with the DNA-dependent DNA polymerase. All right, it's better to write it down, isn't it? Okay, you guys. So now let's flip over to the last page because, folks, we want to talk about some kind of special viruses that can cause kind of special infections, right? So let me clear the board here. So what we first want to talk about, you guys, is some viruses can cause what are called latent infections. And in my mind, you guys, I'm always trying to translate these things. It's like, well, what does that mean? So I think of latent infections as being lifelong infections. And we'll use some examples to try to give you an idea of what we mean. So on your handout, folks, there, it says latency, these lifelong infections. And what happens is you'll get infected. Sometimes you'll have clinical signs and symptoms, and then they clear up. You feel better. You're like, whew, I'm so glad that was over, right? But what you don't know is you're still infected. You've got this dormant infection where the virus is still present in your cells, but it's not replicating. So you don't have any clinical signs and symptoms. But the bad news is, you guys, later, what could happen? The virus could be triggered to start replicating again, and then you'll have another round of clinical signs and symptoms. It's not a new infection. It's a reactivation of these dormant latent infections. Okay, so just some terminology. folks here. So we have the primary infection is when you first get infected, right? Ooh, that sounds like viral music, doesn't it? I just see the shower of little viruses coming down. Okay, primary infection, first infected with the virus, Okay. Okay, and you may have clinical signs and symptoms. And then you're going to feel better. And you think, I'm cured, right? But what we don't know is the virus has infected cells and is dormant, meaning it's not replicating. It's still capable of replicating, but it's just kind of resting, just kind of hanging out, dormant. Okay, meaning not replicating. And if it's not replicating, you guys, you won't have any clinical signs and symptoms. So you think, I'm fine, I'm fine. No clinical signs and symptoms. Nor symptoms. But then, folks, and some of this is still a mystery, there can be a trigger that will reactivate the virus. So we're going to call this reactivation. It's not a second infection. It's a reactivation of this original primary infection. So reactivation, some trigger, and I'll put it a trigger, and again, folks, not all the we don't understand all the trigger. A trigger causes virus to start replicating again. And what are we going to experience? Another round of clinical signs and symptoms. Right? And so this then is going to cause more clinical signs and symptoms. And folks will use the herpes viruses because they're just like the best examples of viruses that cause these latent infections. And so for the herpes viruses, folks, it's a big family. We'll call it the herpes virus family. And there are some herpes viruses, you guys, I would like you to know for the lecture exam 3 because they cause these latent infections. So the first one that you guys might be surprised about is the chickenpox virus. So I know most of you were probably vaccinated against chickenpox, but before the vaccine developed, most of us got infected as kids, right? And if you got infected as a kid, were you permanently infected? Yeah, you're permanently infected for the rest of your life, right? So the primary infection was called chickenpox, the first time you got infected. But then, right, it became lifelong infection. And we know with chickenpox, at least you guys, we know that anything that suppresses your immune system, and for a lot of it, it's just getting old, right? As we age, our immune system starts to fail us. But in young people, you guys, chronic stress is immunosuppressive, and that can trigger reactivation of chickenpox virus. And what do we call a reactivation of chickenpox virus? You got it. And is that ever painful? Yeah, it is so painful, you guys. Right? So it's not a new infection, it's just a reactivation of that primary infection with chickenpox. Now the fancy name, you guys, for chickenpox shingles, it's called the varicella zoster virus. And varicella referring to chickenpox, the first infection, right? Zoster referring to the reactivation leading to shingles. Okay, so you guys all right with that one? Okay. And again, you guys, I know you get mad at me. So if you want some more details on some of these herpes viruses, the blue supplemental sheet, you guys, it gives you more details. I won't ask you anything that we don't talk about in lecture, but a lot of people are fascinated by the herpes viruses, so I wanted to give you some more information here, okay? And then, folks, the another one I would like you to know for the exam This is another herpes virus. Okay, another herpes virus, you guys. Let's see here. Where should I put this guy? More herpes viruses. In the old days, this second group of herpes viruses, they were referred to as the herpes simplex viruses. And I still often call them that because that's how I learned about them. Nowadays, you guys, what they try to do is they try to give numbers to all the different herpes viruses. So these guys are called the human herpes viruses, human herpes viruses type 1 and type 2. So 2 different genetic strains, right? And the human herpes viruses type 1 and type 2, this is one, I think we mentioned this before, you guys. Let me fast forward, see if I have a cartoon of it. Oh, yeah, okay, good. Okay. So with the human herpes virus type 1, if they read the textbooks, they would know they should be causing infections where? Above the belt. And that means mouth, face, really bad news, you guys, eyes can lead to blindness, right? And those of you going into dentistry, dental hygiene, and occupational therapy is what? Herpes infection of your fingers. Why? A person like me, you guys, I know you won't want to get near me, right? But I've got human herpes virus type 1, lifelong infection. Right now, they're hanging out up here in my trigeminal ganglion, waiting for me to get stressed out, right? And so the bad news is, you guys, I can reactivate, I can start shedding viruses from my mouth and not be aware of it. There might be a couple of days where I'm shedding virus in my saliva, right? And I don't know it. So I go to have my teeth cleaned, right? And the dental hygienist is cleaning my teeth with those really sharp instruments. They're gloved up, of course, right? But would it be possible to slip with a tool and pierce your glove and pierce your skin? Yeah, and if I'm shedding herpes in my saliva and it inoculates your finger, now you're infected. And folks, this is called herpetic whitlow, and it's an occupational hazard of folks that work in dentistry. And again, where are you getting infected? From people like me that are shedding the virus, right? Okay, now what the heck's going on with these herpes viruses? So what they do, folks, they initially replicate in our epithelial cells, like epithelial cells of your skin, the mucous membrane. They cause a lytic infection. And so you get these horribly painful fluid-filled vesicles chock full of virus, right? Very painful. And what we don't know is they are also invading the sensory neuron axons. Why are our lips so sensitive? All these sensory neurons. Why are our fingers so sensitive? All these sensory neurons. And you guys are going to talk about type 2. Why is the genital tract so sensitive? All these sensory neurons, right? So what the herpes virus do, they replicate in the epithelial cells and then invade the sensory neurons and travel up the axons. It's like a little freeway for them. And then they hang out in what's called the neuron cell body. That's where the nucleus of the neuron is, right? So right now, you guys, my herpes are hanging out my trigeminal going, just waiting. We're just waiting for you to get really stressed. Usually I get really stressed right before the semester starts. So I start the semester with this great big herpes sore on my lips. And the students are like, we don't know about this person, right? But anyway, it's a great way to introduce you to microbiology. Yeah, okay. So and again, folks, if any of you have had these are so painful. Oh my gosh, they're so painful. And again, folks, I got infected in college. I've probably had 30 reactivations, right? So it's like, and the older I get, right, the more activation I'm gonna get. So it's just not fun, right? Yeah.
You said that people can get warts on their fingers as well?
Not okay. So warts is a different virus. This is a herpes virus, right?
So if a person has herpes and one of their symptoms is warts, is that what kind of type is that?
If a person, so the difference is with herpes virus, they cause a lytic infection, so they're lysing your cells and you get these fluid-filled, painful vesicles. HPV, they invade the cells and trigger your cells to they can't stop growing. So with a wart, it's a solid clump of cells. They're cells growing out of control. So you can have herpes and HPV. I have that too, right? But they're two different viruses, two different infections, two different types of clinical signs. Okay, all right. Okay, folks, so now, the second type of herpes viruses, you guys, the human herpes virus type 2, if they read the books, they would know they're supposed to cause infections below the belt. So transmission there is usually sexual, right? These are envelope viruses, you guys. They're wussy viruses, right? You damage the envelope, right? So that's why they need intimate contact, right? For the viruses to be transferred. And sexual contact, you guys, that's about as intimate as it gets, right? So the bad news with genital herpes is the same story, you guys. Your sensory neurons get infected. They travel up the axons of the sensory neurons, and they're hanging out in the ganglion, the sacral ganglion, right? And it's the same scenario. When you get stressed out, right, you reactivate. And again, you guys, the really important thing for us to know, for us to teach our patients, is we can be shedding herpes virus. We can reactivate and be shedding herpes virus from the genital tract without having any clinical signs or symptoms. And the reason that's important is that the person with whom you're intimate, you want to protect them, right? So you don't want to presume, oh, I'm not shedding, I don't have any burning or, any clinical signs or symptoms. You always want to try to protect the partner with whom you're being intimate, right? That's just really important. Now, one of your colleagues, this was a few years ago, it helped me so much, you guys, because I learned as much from you as vice versa. So she said she had genital herpes, and the doctor had her on acyclovir, and it was helping tremendously. And what it was doing, it was reducing the number of reactivation. It doesn't cure you guys, but it reduces the number of reactivation. It reduces the number of viruses you're shedding, and therefore, if you're being intimate, right, it's going to reduce the chance of your partner getting infected. And furthermore, you guys, one of our concerns with moms who have genital herpes, and it's probably, okay, so to back up, you guys, the patients we're really worried about are pregnant moms that have recently become infected with genital herpes. And the reason is they haven't had a chance to make antibodies that they can pass across the placenta to their baby. Right? So if mom has had genital herpes for a while, she's probably got some decent antibodies, right, circulating. She's going to pass those to her baby so that if she has a vaginal delivery and she's shedding, the baby gets infected, at least the baby has some of mom's protective antibodies on board to protect the baby, right? But if mom has recently got infected, right, and is pregnant, she hasn't had a chance to make antibodies to pass to her baby. So if the baby is delivered vaginally, right, and if mom's shedding, the baby's getting coated in the viruses, the baby's immune system, you guys, is way underdeveloped. So the viruses can spread and cause really severe neurological damage. They can even cause the baby to die, right? So that's like one of our concerns. But you guys, our students have done the research, and what the students said was that acyclovir is safe to give to pregnant moms. And so again, you guys, not going to cure mom, but it's going to reduce the replication of the virus, reduce the chance mom will be shedding at the time of vaginal delivery, reduce the chance of the baby getting infected. In the old days, and I know in some hospitals this is the same way, you guys, and this is your colleagues telling me, one of your colleagues told me she worked at Kaiser, and she said it was their policy that if mom had genital herpes, that they would automatically do a C-section, right? They wouldn't even risk a vaginal delivery. But then there's other folks that have said that's not really universally true, right? The parents can work with the physician, can screen for the herpes, and in some instances, the vaginal delivery will be a safe delivery, right? But you just need to be aware of talking with the doctor and screening for the herpes. Okay, so, you guys, so let's see here. I think, yeah, what we'll do, folks, what day is today? Wednesday, right? So you guys will do a little bit more of this virus introduction. I'll have to check our schedule. so you guys, if we're running too far behind, I'm gonna cut and slash some of the viruses. I apologize, but I don't want us to get too far behind. So we'll definitely have viruses, you guys, on, no, not Monday. Not Monday, you're not gonna be here Monday, right? Wednesday, you guys, we'll try to finish up viruses, and then we're gonna do a little bit on prions, okay? And then we'll just move on to the next unit. We're almost finished, okay? All right, so yeah, so you guys take care.
Do we still have lab for Wednesday before
Yeah.
Thanksgiving?
Oh, sorry, yeah, no.
No.
We'll have lecture, but no lab.
No lab, okay.
Yeah, thank you so much.
So I've had chicken pox before. And
You
I,
have or have not?
I have, and
Okay.
I work in the medical field, so I always have to get the varicella titer to
Yeah.
make sure that I
Yeah.
take to my employer.
Yeah.
So you're saying if I get stressed out enough that I can reactivate that and get the shingles?
Well, if you get the, okay, now, and I had this conversation with another of your colleagues, a young person, she had chicken pox, she's had shingles, she's reactivated. And I said, well, can you look into getting the shingles vaccine? Because this old folks can get the
So
shingles vaccine.
she had it when she was younger and reactivated the shingles as an adult.
And we've had other young students reactivate with shingles.
And she said it's because she was under a lot of stress. For real? That's
Yes.
scary.
Yeah. So what I asked her, and she works at a hospital, she said they won't vaccinate me. You have to be so old. I go, wait a minute. You've already reactivated. You've already had shingles, and they're
Yeah.
not going to vaccinate you.
Yeah.
So I asked her to ask them why,
Weird.
because it doesn't make sense to
Yeah.
me. If you've already reactivated, why not be vaccinated? The logic
That's good for me
escapes
to
me.
know. I have to be careful because I'd be under a lot of stress, and I didn't know
You
that.
all are under a lot
I
of
didn't
stress.
know that. Like,
Yeah.
that could happen.
Yeah, and it might present a student, one of the students that presented, it was right at this time a couple years ago, she presented and it looked like herpetic whitlow. It looked like the finger, like there, but it wasn't herpetic whitlow, it was shingles. She reactivated on her fingers. So it can react, a lot of times people say it's across the chest,
Yeah.
but it can reactivate anywhere. So she activated on her fingers.
All over?
Just one,
Oh,
just a
just
big
one.
vesicle, but
Okay.
it was shingles.
Interesting.
Yeah, so find out,
Yeah.
it's like, why is it that I can't get vaccinated for shingles?
Interesting.
I mean, because young people get shingles.
Yeah.
It's not as common as old folks.
Right.
Yeah.
Yeah, my next I think was 28 got shingles.
We
Yeah.
had
I was
one.
just telling her I had chicken pox before, and then I didn't know that if I'm under a lot of stress, like she just said, I can reactivate.
You can reactivate, yeah,
But she, the root is that.
and it's so painful.
Was he under a lot of stress when he got it? Okay, thank you.
Yeah. Check it out, will ya? Okay.
He was under a lot of stress when he reactivated?
The reason I asked about the warts is because my mom has herpes virus, and
She has herpes.
she has herpes, and she
Most
has
of us do.
warts on her fingers, and she said it's caused by the herpes, and that's why I was
But
confused.
a lot of people when they have herpes, either like type 1 or type 2, they don't have symptoms. So a lot of the time,
Oh, she has symptoms.
a lot of the times
So
they
that's
don't know that they even have
Yeah.
herpes.
So let's back up. So are they true warts?
because
I mean,
yeah,
are they
like
solid?
they're
Okay,
real.
so she
Yeah.
has two latent infections. She's got an
I need
HPV
to tell her about
infection
that. I don't think
and
she
a
knows
herpes
about that.
infection. Yeah,
Okay.
because that's what I have. I've got a latent HPV infection
Mhm.
here, and the wart grows and then it goes away.
She,
And then it
yeah,
grows
she
and
has
it goes
them
away.
all the time. Like they never go away.
They never
like,
go
ever.
away. Yeah.
And she also has, oh my gosh, I just forgot about it. Sjogren's disease.